Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on implantation. In the previous lectures on cleavage and blastocyst formation, we had learned that after the process of fertilization, after the duration of 72 hours, that is 3 days, a solid ball structure is formed, and this structure, which is having 16 or more cell, is known as morula. This morula is moving forward in the uterine tube during day 4 and day 5. During this journey of morula, the mitotic cell division is taking place, due to which the cell number increases and the cell number reaches close to 100. So this 100 cell structure, as we know now, is known as blastocyst. So after day 5, this blastocyst reach into uterine cavity. Now this blastocyst floats in the uterine cavity for two days, and after this 2 day time period, this blastocyst starts to implant on the inner wall of uterus, that is the endometrium. So I am showing you this. So this is the first point, okay, that we have discussed. Here you can see this one is the blastocyst, and this is the endometrium of the uterus. So we can say this blastocyst is just touching the endometrium. Now, which cells are just touching the endometrium? So the trophoblast cell, which are present over the inner cell mass, they are adhere to endometrium. So we can say, in other words, the trophoblast are the cell which are touching the endometrium. So as you can see here, these are the trophoblast cell, and they are just adhering, they are just touching the endometrium. Now what this trophoblast cells are doing? So this trophoblast cell, they start secreting digestive enzyme, and they also secrete growth factors onto the endometrial surface. So as you can see with the color dots, I am showing that this trophoblast are secreting the digestive enzyme and the growth factor on the endometrium. So this was the second point. Now we are moving to the third point. So this is the third point. This is the endometrium. Now what is happening to endometrium when the implantation begin? So the endometrium quickly thicken, okay, and it is showing the characteristic of an acute inflammatory response. So we can say, in other words, the endometrium is proliferating, it is swelling, it is increasing in size. Okay, so this is the third point, that the endometrium is quickly thickening and it is showing the acute inflammatory response. Now, in the endometrium, at the time of implantation, there are uterine blood vessels. As you can see with the red arrow, I am showing this blood vessels. These blood vessels are shown in the cross section, and these blood vessels becomes more permeable and leaky. So which are the cells that leaks out from this blood? So as you can see, the inflammatory cells, that includes the lymphocytes, natural killer cells, and macrophages. These cells comes out from the blood. They invade the area. So from this blood, these three type of cells, they are coming out from the blood. So this is the fourth point you need to remember. Okay, now we are moving forward on the fifth point. Now these are the, in this figure, just look at this topoblast cell. This is the topoblast cell. Okay, and here this topoblast cell, for example this layer, this topoblast cell, they start proliferating. That means here the topoblast cell, they start divide mitotically. So many cells are produced by this cell division, and these cells are arranged in two layers. Okay, so this layer, we can say the outer layer, and this layer we can say is the inner cellular layer. Okay, so this is our fifth point. As you can see, the topoblast is proliferating now, and it is producing two distinct cellular layers. So the cells which are present inside, that is the inner cellular layer, is known as cytotopoblast or cellular topoblast. And these cells, cytotopoblast, or which are known as cellular trophoblast, they are having the cell boundaries. That means these cells, they retain their plasma membrane, and the outer layer cell, they are known as syncytiotropoblast, okay, or also known as syncytial tropoblast. This cell loses their plasma membrane. So I am showing you in the figure, so you will get the idea. We are looking at two layers. One is cytotrophoblast and another one is syncytiotropoblast, and both this cellular layer, they originate. So you can see this thing, just focus on this thing. Okay, this is a tropoblast, and this layer is start dividing. So this layer and this layer, this two layer, I am zooming this. Okay, so this layer, okay, is known as syncytiotropoblast, and this one, this layer, is known as cytotropoblast. So this, yes, they are having cell boundaries. These cells are having cell boundary. Okay, that means they are having plasma membrane, and in this cell, okay, in these cells, the plasma membrane ruptures. So now I am showing you one cell. Okay, this is the syncytiotropoblast, for example. So what happens? This plasma membrane is eroded. The plasma membrane ruptures. So all the cytoplasm of the cell and the nucleus will come out. That is the thing. So this cytoplasmic mass with the nucleus is known as syncytiotropoblast. So you need to understand that all this cell, they are losing their plasma membrane. Cytoplasm along with the nucleus, is coming out, and this entire multinuclear cytoplasmic mass is known as syncytiotopoblast, and this is the outer layer they are producing. So again I'm showing you the another figure so that you will get the idea about this. For example, over here you will get the better view. So here, as you can see, you closely observe the cell, the syncytiotopoblast. Okay, now you see this is the plasma membrane and it is going to rupture. So cytoplasm is coming out and the nucleus. So this multinuclear mass of cytoplasm is known as syncytiotopoblast, and this cellular layer is known as cytotopoblast. So now you get the idea of these two cellular layers. Okay, so we were on point number 5. Now we are moving on the point number 6. Now you see what is happening. You focus on this syncytial region. I am showing you in the figure also. Can you see this? This syncytial region is protruding now. Okay, for example, this one is syncytial region, and it is making large protrusion. So what is this? What is this syncytial region? Actually it is a cytoplasm. Okay, and this cytoplasm is having enzymes. So these enzymes are digesting whatever the material of the endometrium comes in the way. All the cells, whether it is a this lamina propria or the uterine gland, they are digestive everything which comes in their way. So we can say that the syncytial region is sending out the long protrusion, and they are digesting, they are invading the endometrium. Okay, and they are getting, by doing so they are performing two function. One, that they are getting nourishment, and second thing, they are making the way for the incoming blastocyst to implant. They are making the way for the blastocyst to implant inside the endometrium. So this is the sixth point. Okay, so you see this protrusion. Okay, so it is in these protrusions, which are cytoplasmic and multinuclear, they are eroding the endometrium. Okay, so now the endometrium is eroded due to this cytoplasm. So we can imagine, for example, if this is the endometrium, this is the blastocyst. Okay, and the syncytial protrusion, they are digesting these cells. So now what happens? Here there is, this entire region is eroded. So here there is a large hole, and this blastocyst fit into this hole. Okay, and this hole or this burrow is like a velveting lining. So for example, this is the eroded endometrium, this is the eroded endometrium, and blastocyst just fit into this eroded endometrium. Okay, now this blastocyst is covered by the pool of blood. I am explaining. For example, over here, as you can see, this is the blastocyst, and around this blastocyst there are blood vessels. So these blood vessels are also ruptured due to the enzyme of this syncytial region or this protrusion. So this blood vessel of the endometrium, they rupture, and the blood fills in the cavity, and these cavities are called as lacuna. So in the figure, as you can see, this is the blood vessel, and the enzyme will digest the wall of this blood vessel, so the blood leak out, and this blood accumulated in this empty space, and this empty space are known as the lacuna. So we can say the lacuna is filled with the maternal blood. So over here in this figure you can see the maternal blood, and the blastocyst, okay, is burrowed. In the burrow, the blastocyst is present. In the burrow or in the dig, this blastocyst has entered. Now, so you can understand the endometrium has invaginated, has inserted blastocyst in inside. Okay. Now, so this was the seventh point. Now blastocyst has entered into, as you can see over here, the blastocyst has entered into, this is the endometrium, and the blastocyst is now present inside the endometrium. So now this endometrium proliferate. Okay, now what is the meaning of proliferating? So now you have to understand this thing. For example, this blastocyst is inside the endometrium, and by that time, due to the syncytial enzyme, this layer was eroded. Okay, this layer was ruptured, and by doing so, this blastocyst had entered into endometrium. Now, since this blastocyst has implanted, again this columnar lining epithelium of columnar cell, they will start dividing again. Okay, and here there is a lamina propria. So this lamina propria, then the uterine gland, and this cellular layer that is known as the lining epithelium, they are covering now this blastocyst. So now we can say the implantation has completed now. And the last point of our today's discussion, that once this embryo has implanted, so in at initial stage the embryo obtains its nutrition by digesting the endometrial cells. We have learned that the syncytial protrusion, that the cytoplasmic enzyme, they were eroding, they were digesting the endometrial cell, and the embryo was getting the nourishment. But by the second month of the pregnancy, the placenta is developed, and now this placenta is providing the nutrients, oxygen to the developing embryo, and the placenta is also performing the function of removing the metabolic waste. Okay, so in the next lecture we will learn about the placenta formation. So one by one, before concluding this lecture, I would like to show you all the slides. Okay, so you need to go through all the figures, and once again you go through all the points. We have started with this. This was the second point. Then this is the third point. Then the fourth point. Then this is the fifth point. Then this is the sixth point. And this is the seventh point. This is the figure. Then this is the point number 8, and this is the final point. So with this, we have completed the discussion of implantation. I hope you have enjoyed this presentation lecture. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.